And the presence of cobalt atom is actually by evidenced by the crystal structure analysis. Okay, and most important part of the contribution of the Dorothy Hodgkin is the structure of the insulin. So she actually she took 3 decades to know the structure of this molecule is today. Okay, I think you all know it has been given. It is actually given to the. So earlier, of course, crystallize this insulin and solve the crystal structure, and because you are getting insulin, the companies are actually making this, preparing this particular drug in the large scale, because we know the structure of the insulin now. Look at the patients of this lady. She almost 30 years she spent on. She started in 1935, and she could solve the structure of the penicillin, so of this insulin in 1969. Who should have that much patience? Okay. And then as everything started, molecular biology started, then the structure of the myoglobin was determined, structure of the hemoglobin was determined, and then the other two giants, genius people, I would say, the John Kendrick and Maxwell, they also received a Nobel Prize in 1962 in the structure. Okay, and then I just he of course set up the molecular biology. Then the structure of the so what is this? Something is happening here. It has gone. Okay. So he set up he was a pioneer in the field of the DNA research, and actually X-ray diffraction image of the development. So the DNA was crystallized by the Rosalind Franklin, and she could record in fact the structural diffraction pattern of the DNA molecule, as you can see here. Okay, but unfortunately she could not interpret this. She not I think she interpreted the structure, but unfortunately she died in a road accident, so she could not propose it. Okay, but later on, you must be knowing about Watson and Crick, who actually solved the crystal structure of the DNA. It's a double helical structure, as you can see here. You have the sugar moiety which actually connect the nucleic bases like this. So it is a double helical structure. So this structure was solved by the Watson and Crick. But they utilize the same photograph taken by the Rosalind Franklin, and they could interpret the structure. And the Nobel Prize is given to Watson and Crick, but unfortunately it was not given to the Rosalind Franklin because she was died, and Nobel Prize not given to the dead people. Okay, so in fact, yeah. So this is a story of the DNA molecule. Both of them got the Nobel Prize in 1962 by proposing the structure of interpreting the structure of that 51 taken by the Rosalind Franklin and giving the structure. Now, we started with the sodium chloride, and then today, what is the limitation of the solving the crystal structure or the crystal structure? This is the structure of the ribosome. Ribosome, is a machine which actually synthesizes the protein in your body. The structure of the ribosome was solved by the Venkataraman Ramakrishnan, Thomas Stage, and Yoda Adayonath. Okay, and you know how many atoms in this structure in this particular ribosome? There are more than 1 lakh atoms are there, so there is no limit now. So we went to the 2 atoms which are in the sodium chloride. Now we can go to the very big molecule, we can say ribosome, which has more than 1 lakh atoms. So there is no limit. So you can solve the structure of the any atoms now. Okay. So the crystallography in a nutshell, you have to have the crystal, and then you have to have the instrument, which actually you have the X-ray source and all. Then export the crystal to the X-ray, you get diffraction pattern. And then, of course, to interpret the data, you need a lot of mathematics there, which I am not discussed here now. Okay, a lot of other things are there, and you can finally get the crystal structure. Okay, so this is the electron density equation. Electron density is nothing but it tells you the density of the electron particular position inside the crystal. Okay, this is the instrument.
Sir, your slides are
you
not.
Can study the Excel. Sorry.
Sir, your slides are not moving, so it is stuck to crystal structure of DNA.
1 minute. 1 minute. 1 minute. 1 minute. No, it is shared. It is not visible.
The slides are
Hello?
not moving. It has stuck to crystal structure of DNA. Like students.
Now it is not moved.
No, it has not moved.
Now go. Hello?
Now
Try move.
also, yeah,
Can I stop shaking and shake once again?
yes, that would be fine.
I'll do it. Okay? Just a minute. Okay. So yeah, so one
Yes,
minute.
sir.
I start sharing the screen my once again. Okay. So can you see my screen?
Yeah, it is clear. So make it.
Yeah. Now I go to slideshow mode.
Yeah.
Is this slide is visible now?
Yeah, now it is visible, sir.
Okay. So now I'll scroll it. Okay. Yeah. In fact, I was in this slide. I was telling here that this is the crystal agape inside the laboratory. You have to have the crystals, or you have to grow the crystals, of course, because every compound, chemical compound, of any compound, biological or protein, anything, you will not get in crystalline form. You have to grow them, and then once you have the crystals, you have to have the laboratory setup for this. You have to expose the crystal with the X-ray and you get diffraction pattern, and then this diffraction pattern you have to interpret. So there is a lot of mathematics. I am not going detail of this, but this is the way you have to interpret it. And at the end, you will get the electron density map. Electron density map in the Timodio crystal structure. Okay, means a molecular structure inside the crystal. This is the particular setup inside the lab. You have the X-ray source from where the X-ray comes. The crystal you mount on this one, and there's a detector here. So there is a microscope through which you can see the crystals and all. And this is a typical setup of the diffractometer axis and all. I will not discuss it much. So actually, our aim is that when you mount the crystal on a diffractometer, each and every piece of the crystal has to be exposed to the X-rays. And because of that, there are different ways you can rotate the crystals. Okay. So this is a design of the particular diffractometer, as you can see here. They rotate at different angles, at different positions. Here, a detector, which also can be moved like this. It can be taken back and forth also, as you can see here. So depending on the external condition, you can always set the position. Okay. So main image here is that we have to expose the each and every part of the crystal to the X-ray. Okay, that's the main. Okay. So this is all about the crystallography and the applications. So now I'll go to the more applications part of it. I will not take more than 15 minutes now. I think within 15 minutes I can wind up this program. Okay. So the polymorphism. So now when we started knowing the structure of the crystal, knowing how the molecules are arranged in a crystal, what are the different ways they can arrange in a crystal. So now we can now correlate the structure with the property. Okay. So first thing is that polymorphism. So what is a polymorphism? I told you about I told you about the carbon electrons. Carbon can arrange in three different ways inside the crystal. One is a diamond, another is a graphite, a third is the polygon. So what is polymorphism now? The ability of a compound to exist in more than one crystal form. Molecule is same, but arrangement in a crystal is different. And because the arrangement in the crystal is different, the properties are. Now let us see this is a cartoon diagram I'm showing here. See, the molecule is same. Only thing, it is again differently. So this is a polymorph 1, polymorph 2. Arrangement is different. And because the other arrangement is different, they have the different properties. Taking one example of the paracetamol. I think you know the paracetamol. Most of you know paracetamol. Whenever you get a fever, you take one tablet of paracetamol. I have only one or two examples. Okay. So now let us see. It exists in two polymorphs. One is called the monoclinic. Again, the molecule has a zigzag arrangement, as you can see here, and orthorhombic, which is a planar or sheet arrangement, as you can see. See, this is the very stable. This is the metastable form. Metastable means it is not stable up to the stable. It converts to the another form. So this is a stable form, but the problem with this form is that it has a zigzag structure. Okay, and because of that, it is difficult to compress. If you want to make the tablet of this, it's difficult to compress. Even if you make the tablet of it with the excipients, it develops cracks after some days, after months, some months, and it develops. Okay, so this is the problem with this one. Orthorhombic. This is easy to compress. You can make a tablet of this, but problem with that, during the storage, this form can go to this form. So this is the problem. So see, earlier it was not known, but after studying the structure, we can. Okay. Now coming back to the chocolate, you all eat chocolate, I know, but do you know that there are 6 polymorphs of the chocolate? There are 6 polymorphs. The cocoa butter can crystallize in a 6 different forms, but only one melts pleasantly in the mouth and has the surface sheen and crisp hardness that makes it so tasty. This tasty crystal form is not very stable, but this is not stable. It actually gets conveyed to the more stable form, and this more stable form is dull. It has a soft texture and melts only slowly in your mouth. Okay, so all these different properties or different taste of the cocoa butter can be explained if you know the structure of it. Okay, so this is all. I don't know whether you know about the HMX. HMX is the high explosive, like RDX. RDX you might have heard, HMX you might not have heard it. But this RDX also exists in different forms. This is the alpha form. This is the structure of the RDX. Okay, alpha form, beta form, gamma, and delta form. And let us look at their structure. See, the structure is different, and because the structure is different, the problems. Otherwise, what will happen if you feel this HMF in the ammunition? It should explode. Sometimes it doesn't explode at all because sometimes these forms form alpha, beta, and gamma, and delta forms comes doing crystallization during the manufacturing process or crystallization process, and because of that, it may explode, it may not explode. This is very unstable form. Sometimes what happens during the transportation of the explosive, it can explode there only. So you have to be very, very careful. So now we have to control our crystallization in such a way that you should only get the beta form, not the alpha and gamma form and the delta form, because these are unstable forms. So during storage or during the transport, they can explode out. So this is the importance of the polymorphism. So how we came to know about this? After knowing the structure only, we came to know, okay, oh, this had this form, had this form. Okay, now let us come back to the colors. Color crystal. Why this color? Why there are colors? And you can see this is the molecule. Why it has so much color? It ranges from the orange. This is from the red color, orange color, yellow color, or different shapes, different plates, something. Why it is so? And this is because, as you can see here, I don't know whether we can see this picture. The molecules have different conformation, different orientation inside the crystal, and that is actually giving rise to different colors. So the color का जो mechanism है, why color is like that? We can answer those things now. Okay, look at this, different colors. As you can, this is the molecule which are different colors: orange color, red color, green color, and something yellowish color. So all these colors is because of a different arrangement of the molecule, different conformation, different orientation of the molecule inside the crystal. This is another example of that. Now coming back to topic called crystal engineering. Now what is crystal engineering? So crystal engineering is something, since till now what I have covered in the class is that now even if you have the crystal, we can solve the crystal structure, means we can determine the molecular arrangement inside the crystal. That means we know how the molecules are coming close to each other in a crystal. There is a specific way the crystal molecules can come in. They form the some bonding within the molecule. Now see, this bonding is not a covalent bonding. This is just something called a weak bonding. We call them the non-covalent interactions. So these are called the molecules inside the crystal are having hydrogen bonding interactions. So the molecule come together by hydrogen bonds, or the halogen bonds, or the pi stacking interactions and all. So what is crystal engineering then? Understanding the molecular interaction. So as I told you, molecule interacts in a specified manner inside the crystal. So if you can understand that in the context of the crystal packing or the molecular arrangement and utilization of such understanding, if you understand this, how I generally call it a molecular psychology. If I understand the psychology of the molecule, how they come close to each other inside the crystal, I can design a crystal of my choice of desired properties. That is called the crystal engineering. Okay, so now then I can make the such setup. I can see this is a dry benzoic acid, isn't it? Tricarboxylate benzoic acid, you can see here. The molecules can again like this nice two-dimensional focus structure, or they can have this structure, or they can have this structure, or you can make the structure. If you can understand how the molecules are coming close together, you can make the choice. You can engineer the molecular arrangement inside the crystal. So you can make this, you can make like this, you can make the adamantine tetracarboxylic acid. They leave the nice structure here. You can design the molecule of your choice. Now, what are the applications of crystal engineering? I mentioned to you what is crystal engineering just now. So you can utilize in the pharmaceutical, making a drug co-crystal. I'll tell you why it is important. Or in solid state reactions, in thermosolvent material, in property modification, MOFs and COPs. I'll discuss what is MOFs and COPs also. Okay. So what is a co-crystal? Co-crystal is nothing but, suppose you have the two molecules, a molecule A and molecule B, or crystal A and crystal B. Now you mix them together, and then what actually happens when they dissolve inside the solution, they form the non-covalent interaction between them. And now you in the crystal you have the molecule A as well as the molecule B, and molecule A and molecule B are interacting by non-covalent interactions. So like this. So this is the carbon one of the drug, and this is a succinic acid. So now see inside the crystal you have both the molecules. Okay, but why we are doing this? This is a theophylline. Why we are doing this? To modify certain properties. Okay, now I took the example of the paracetamol. I said in the paracetamol, if you get the most thermodynamic crystal, most stable crystal has a zigzag packing, and because it is a zigzag packing, it was difficult to compress. So then what actually would can be done? Can we make the co-crystal of the paracetamol with the oxalic acid and see the packing? Now see, this is the structure of the oxalic acid and the paracetamol. Now they have this structure. So I can engineer the arrangement within the crystal, within the crystal. So now it has a sheet structure. It is more stable compound, more stable crystal now. So the concept, the stability issue was taken care of, as well as the issue with the issue with the compressibility has also been taken care of. So if you can see here, this is the actual pure paracetamol. Like that with the oxalic acid, you can actually make a sheet structure, and then the tablet which will actually breaking or fragmenting actually stable now. If you make the co-crystal of this. Okay, so this is one of the application. Second application I will tell you. So this is a caffeine which is very hygroscopic in nature. It picks up the water from the atmosphere and it becomes a hydrous. And because of that, it is very unstable. Okay, so now if I co-crystallize this caffeine with the like oxalic acid, malonic acid, or succinic or butyric acid, I can co-crystallize them together. So you can see this is a caffeine, this is a oxalic acid. It has been made. Now you can see this is very stable compound now. It doesn't take the water from the atmosphere. So at this relative humidity 98% also, it is stable up to the 7 weeks. This is the crystal structure with the malonic acid. You can see it is stable up to at 98 humidity. It is stable up to I can say 7 up to 75. It is stable up to the 7 weeks and all. But later one up to it takes the water. Okay. So with the co-crystallization technique, we can modify we can alter the properties of the crystal. Let us look at this here. This is one of the example. See here, this is a furosemide, another drug molecule, and this is a bipyridine. See, both are colorless. But when you make the co-crystal, they develop the different crystals. So different polymorphs you can see. So we can make the color crystal also. Here, see, look at the colors. And this is colorless. Okay. We can also make the fluorescent crystals. See, if you have the crystals, so this is a fluorescent crystal. So when you co-crystallize this molecule with any of these molecules, the crystal can actually become it can reflect blue light as well as the red light depending on which molecule you are taking. So now see, this is AD. AD is nothing but ADs and ADs and this is the molecule. Okay, so if you can co-crystallize this with this one, I can get this crystal, and the crystal can be in blue color. If I want red color, I simply have to co-crystallize this with this cyano. I got a red color, and this is cyano. So this is my red color. So I can cover the full entire range of the visible light, isn't it? I can go from the blue to the orange to the yellow to the red. So blue, green, orange, yellow, and red. So all these things can be done. Okay. So lot of applications of this technology in the material science, in fact. Okay. Now, can I explain the jumping crystal? I don't know whether you heard about this name, but what actually happens if you some crystal it happens. Okay, not for all the crystals. If you hit them, they jump. See, can you see this? The crystals are jumping. See, it doesn't happen with every crystals, but it can happen with some crystals. So, can I explain this phenomenon? Yes, I can explain. If I know the crystal structure, I can explain the phenomenon. How do I do? See, this is the crystal with, at some temperature. Okay, so when I hit this crystal, as you can see, there are two benzene rings here. The distance between the benzene ring is very high, but at particular temperature, they come close to each other. So when they come close to each other, they release the excess energy. Okay, and then because of that excess energy they release, the crystal jumps. So we could actually find out why the crystal jumps by knowing the motion of the molecule or the before the jumping and after the jumping. So all these things can also be explained based on this technique. Okay. Now coming to the porous crystalline material. So as I said, we can engineer the crystals of our choice. So we can make the porous materials. So porous material can be made on the organic linker and metal ions. So this is one dimension porous material, two dimension, three dimension material. And then these porous materials are very, very helpful. These are the crystals of the porous material for many applications. Okay. So I'll just show you one example of what is the application of the porous material. I hope you can see this video. So as you can see, these are the porous material which have been engineered for our purpose. I want this porous material to have a three-dimensional porous network, and then within their porosity they can absorb the gases. So I hope you can see this. So this is a porous three-dimensional porous network of the metal organic framework, and now you can utilize this metal organic framework for the absorption of the gases. So we can absorb the gases like nitrogen, carbon dioxide, oxygen, hydrogen gas. So I can engineer and then it's called a covalent organic fiber. In previous case, I showed you so metal organic fiber. There was a metal inside it. Now there is no metal here. Now the molecules are arranged. So this is one molecule, is another molecule. If I can then crystallize them together, so I can call them a co-crystal. They can find a nice porous network like this. But now here, the molecules are connected by the covalent bond, not by the non-covalent bond. Okay, and then I can make the material of such porous material which can be utilized for different, as I said you, for gas absorption, for catalysis, for gas separation, for many, maybe it has a lot of applications. Okay. And then last part, drug design of my talk. So can I design a drug? Yes, we can design a drug because I know if I know the target where the drug has to go and bind, and then I should know the structure of the target. So all the drugs which have been, you can see they are mostly the anti-cancer drugs. This actually has been designed based on the structure of the target molecule. So if I know the target molecule, I can do the docking studies or the binding studies. Theoretical binding studies can be done. Okay. So then I can say that. So that should be in this case, this is a nilotinib which actually binds to the target, shown by this purple color. It actually binds to the target of the cell, which actually we have to stop. See, in the cancer, we have to stop the growth of that cancerous cell. So we have to design a molecule which actually binds to the active site of the target cell, so that its growth can be stopped. So we can design the inhibitor. So this are all inhibitors. Okay. Taking another example, here you can see here JPT with binding to the cancerous active site of the cancer cell. So because of that, one can stop the growth of that cancerous cell. Okay, so see the job. So unless I know the structure of the protein of the target cell, how can I design the molecule? So everything. So this structure actually came from the crystal structure analysis. Ritonavir, it's an anti-HIV drug. how it binds to the active site is shown here. Sunitinib, another anti-cancer drug, sitacaprotine, it's one of the diabetic drug. How it actually interacts with the body enzyme can be is shown here. So all these things, all this study you can do it if you know the structure of the compound. All of you. So now with the help of the crystallography, we know the proteins. We can solve the structure of the proteins, DNA, sugars. So then we can see how the way the protein folds inside your folds. So then the structure function relationship also can be calculated now. We know the structure of the polar regions, lipids, and all. So now there is nothing is unknown. If you have the crystals of these compounds, the structure can be determined and the properties can be correlated. Okay. The last two slides of my things. So now coming back to the COVID-19, lot of research is going on this COVID-19 disease. So see the many of the currently what is happening, many of the drugs which are there in the market are repurposed. You might be knowing about, okay, because for developing a new drug, it takes minimum like 10 years. If you want to develop the new drugs, okay, and you have to spend almost $2 billion rupees. $2 billion, not rupees. So then, unless you know the structure of the target cell and the spike protein of this COVID coronavirus, how are you going to design a drug? So now the structure of this target cell of the ACA2 is the target cell in your body, and this is the structure of the spike protein of the coronavirus is determined, and this is the binding site, as you can see here, RBM, being the receptor binding motif. So here the spike protein actually binds to the target receptor of your body. Okay, the same structure portrayed by the one individual. So this is that. Okay. So the structure was determined. You can see this recently it has been published now. So now we have to the drug companies or the researchers or the scientists have to develop the new drug which can actually inhibit this binding. Okay. So if we start inhibiting the binding, then the drug will come in the market. So this is the way. So see the target molecule as you can see here. So all the molecules are shown here. This is also recently published. So this actually the 13B. So this is the structure of your SARS-CoV. You can see here SARS-CoV-2 main protease. So now if you so these are the drug molecules which actually bind to the active site of that coronavirus main protease. You can stop the growth of these viruses in your body. Okay? So these are the recent developments. And the utilization of the crystallography by knowing the crystal structure of the target SARS-CoV-19 as well as we can find the inhibitor which can stop the growth of medicines. Okay, so see, I'm going to end now. So we started with the Lawrence and William Block. They gave the structure of the sodium chloride in 1913. And now we know the structure of the ribosome, which is almost thousands or lakhs of atoms inside the crystal. Okay. So this is the journey, and there is no ending now. Any of the compounds can be crystallized, and then the structure can be solved. So in 2014, we actually celebrated the International Union of Crystallography. Of course, to honor the 100 years of X-ray diffraction by Professor Lavi. Dr. Professor Desiraju was the president at that time. And then this was we had a International Union of Crystallography seminar and congress in 2017 at Hyderabad, and this was the largest crystal structure model of the sodium chloride was built. Was actually was kept there. In fact, it was from Vienna to Hyderabad. It was built in Hyderabad and Vienna, and then got it in Hyderabad. Okay, so this was all there. And then during that period also we have also some booklets on the crystallography. Crystallography matters was also published. It was in all the original language and the English was also published, and visually some of the stamps also were published at that time. So as you can see, in India we had this is a curcumin structure. So stamp on the 2014. Sorry, so this is for the in 2017 or 14. I don't remember now, but it has been published. So you can see the structure of the curcumin stamp launched by India. Other countries also launched the stamps. So you can see it. Okay. And this is the list of the Nobel Prize winners who worked in this field of execution crystallography. I had a total 33. I don't know how many are there in this list, but huge. This is the only field where a huge number of Nobel Prize has been given who actually really worked in this in the field of structure determination using X diffraction technique. So I thank you all for your patient hearing. I'll be happy to answer the questions now. Thank you. So Dr. Pooja, I finished my talk. If there are any questions, I can take those.
Yeah. Can you hear me?
Dr. Pooja, are we around?
Yeah. Can you hear me?
Dr. Pooja? Yeah. Anyway,
Sir, she has not yet unmuted.
yeah. Any of you, the students, have any questions? I can answer those. Students, are there any questions to be answered?
Sir, can I ask a question?
Yeah. I'm here to listen to you now. You have listened me. I'm here. Okay.
Yes, sir. I have a question. Sir, is magnetism an intrinsic property of crystals?
Yeah, it's interesting property of the crystal. Okay. Yeah, please go ahead.
Hello. बढ़िया कपिल.
X-ray strikes the metal, X-ray actually goes inside the atom.
वापस करने हैं बचे हुए bills.
Okay, and actually the X-ray repels innermost electron from the atom. So X-ray hits the atom, dislodges the innermost electron from the shell, and then because there is a vacancy in the innermost shell of the atom, that vacancy is getting filled by the uppermost electron. So the uppermost electron in the shell, electrons from the uppermost shell transit first to the innermost shell. So this transition gives rise to energy, and that is your X-ray.
Sir, understood. Thank you, sir.
Okay, welcome.
Sir,
Yeah, go ahead. Kindly identify yourself. Okay, Rudra Singh. Rudra, please go ahead.
No, sir, I'm Smriti.
Okay, Smriti, go ahead.
Can crystallography determine the presence of crystals in a given area for extraction or mining? Sir, if through crystallization,
Yes.
can we determine the presence of crystals in an area for mining?
Yeah, of course. If the crystals are there, we can of course find out where are they. Okay, no issue.
Okay, sir. Thank you, sir.
Yeah.
Excuse me, sir.
Yeah, go ahead.
Sir, it's Pranav here.
Yeah, go ahead, Pranav. Yes.
Yes, sir. So, like, sir, the crystals are packed in a particular regular shape, right? So can it be a
Yes.
circle? Can it be a
Hello?
circle?
Yeah, it can be spherical in shape also.
It can
See,
be.
the spherical shape depends upon the how much pressure is there. It also see why the water bubble is spherical in shape. It all depends upon the external forces. Okay, so crystal can be in the spherical shape also.
Okay, sir. Thank you, sir.
But the crystal system will be 7 only. Okay, they will be belonging to the 7 crystal system only. Either of those. Yes, go ahead.
सर सर आप There are all motifs of the same size.
No, I didn't get question. Who is asking?
Are all the motifs of same size?
No, I think Mrigank is speaking, but I'm not able to hear him properly.
So students, I am back. I had some network issue. Sorry for that. So you can put your question in chat so that I can moderate.
You can also put the question in the chat box. Okay. Are there any questions in chat box?
Yeah, there are questions, sir. So I'll take few questions.
Yeah, please go ahead, ma'am. Okay.
First question is from Kotha Satvik. If we can engineer the structure of a crystal, why can't we create a crystal harder than diamond?
See, the diamond was created at the huge pressure. Okay, developing that pressure in the laboratory is not possible at all. Okay, the very high temperature and very huge high pressure, the diamond what crystal was created. Okay. Even if today we cannot manufacture diamond as such. Okay, so the laboratory condition doesn't allow you to do that much pressure and that temperature, and because of that, you cannot do it.
Right.
Okay.
Right. So the second question is from Kiran. He's asking
Yeah.
about the solid metal of different crystalline structure because they exhibit different properties.
I didn't get the questions. Kindly.
Are these all solid metal are of crystalline structure only because they exhibit?
Yes. All the solid metals are crystals. All solid metals are crystal, and because they are crystal, they are solid. Okay.
Okay. Then another.
They have the regular and the periodic arrangement.
Right, sir.
All the atoms in a metal have the regular arrangement.
Right, sir.
Okay. Only thing because the electrons inside them in the metallic atoms are free. They are not bound to the only one of the atom. Okay. And because of that, they shine. Okay.
Right. So next is how do we determine impurities in a crystal?
See the impurity in a crystal. If the crystals are doped, we can determine that. But I would say when a compound is crystallized, it is the purest form. It cannot have the impurities. Okay, so crystal is always pure. That's why they are so shining. Okay, so if you are so crystallization is a process of developing the crystals. So even if you have the impurities, simply dissolve the compound in some solvent and develop the crystals. So inside the crystal, impurity will not go at all. If it is going, we can find the crystal by using the crystallography. We can find out whether what is that impurity also. Okay. Suppose some of the if you dissolve some compounds in a water, the water molecules get trapped inside the crystal. So you can find out the presence of the water molecule inside the crystal if you determine the crystal structure.
So next.
Yeah, I think I answered the question.
Yeah. The next question is ferromagnetism an intrinsic property of a crystal?
Yeah, just now I answered that question.
Okay, fine.
You were not there.
Okay. So we will take now.
Dr. Puja, answer that question.
Yes, sir. We will take last question from Nishank. He said it is lustrous and it is a form of carbon, but carbon is not lustrous. Can you comment?
So the luster comes because of the arrangement of the molecules, carbon atoms inside the crystal. So if you see the carbon atom, of course, is not the property of that particular lusterness or softiness or the shining, whatever comes because of the arrangement of these atoms inside the crystal. So the proper crystal structure, arrangement of the molecules. Okay.
Right. So can you lastly comment about scope in the field of crystallography?
Sorry.
What is the scope in the field of crystallography?
Yeah, I didn't get the question. Hello.
Sir, what is the scope in the field of crystallography? This is the last question.
As I told, a lot of applications. You go from biology to the chemistry to the physics to the material science. Lot of scope is there. Because you know the crystal structure determination, you know the molecular psychology, how the molecule pack inside the crystal. So you can design the crystal of your choice. Either it is a biology, protein molecule, or any other molecule. It's called a protein engineering also has come up. So you can do that. The scope is everywhere. Lot of scope is there. Only thing you have to be in science. Okay.
Thank you very much. So I thank you, sir, for very nice and very detailed talk covering all topics related to crystallography, how it can be used in different disciplines, and answering all the questions, clarifying all the queries. I also thank all the students for being with us. I would like to inform the students that we will meet again at the same platform during the Media Site EL webinar session. Until then, thank you. Take care. Bye. Thank you, sir.
Yeah. Thank you, students. Thank you all. Stay home. Stay safe.
Thank you, sir.
Okay. And enjoy your school. Okay. Online school. Whatever it is. Okay. See you then. Okay. Have a nice day. Bye. Bye.
Thank you, sir.